Let's move on to the another important mark, second mark, histone 3 lysine 9. Okay, so the first one is gene activation mark, which we talked about this one. The second is this one. Okay, this is activation, this is repressive, and then K27, this is repressive, which I am going to explain. This is repressive, repressive, on histone 3. So, this is the histone 3 tail modification, the 4 modification, a methylation mark, which I am going to explain. 3 are repressive epigenetic functions, and then 1 which we discussed now, it's a activation functions. Very important mark for the cell differentiations. It works together in a cell differentiation wherever the suppressed genes need to be activated. This protein play very important role in introducing the trimethylation mark. Right, coming to the trimethylation mark at these positions, as I said, this is a repressive mark, very important gene repressive mark, a epigenetic mark on the histone 3 proteins. So, like other many other positions, this also undergoes for the mono and di and trimethylation mark. See, the K4 either 0, in this case H3 K4 Me0 is a well defined function. Me0 is repressive, right? And then opposite H3 K4 Me3 is a activation mark. So, the intermediate mark is not well defined. Okay, so that part is not very clear. So, as of now, the literature or scientific information available only about Me0 or Me3, but obviously it generates intermediate mark. That's what I explained using the mechanisms. But where is the K9? There are studies that confirm the intermediate marks, particularly K9 Me2 play very important role in a specific genes, and Me3. Okay, so each and every mark localization they perform, it's a significantly differ, but in terms of gene activity or regulating gene functions, Me2 characterized well and Me3 also characterized very well. So, by default you should consider now Me3 is a repressive mark, and this also a repressive mark. Okay, so the localization studies for the mark, initial times they were trying to localize. How do they distribute in the cells these marks using the antibody? They found this Me1 something like small punctate domain within the nucleus, whereas the Me2 largely in the nuclear periphery, and then Me3 obviously in the pericentric heterochromatin. Repressive mark largely in the condensed region it present. So, initially they optimized using this BRDU labeling, because this is the thymidine analog which is commonly used to measure the active cell proliferations, active cell divisions. Particularly if you want to study, BRD staining can be used. You had a BRDU to the cells, and then this will get integrated into the newly replicating cells because of thymidine analog. Then you can use the antibody against the BRDU, and then you can localize or pull down and look for a functions. The BRD staining, this is a stage at different cell cycle, and then they connect along with the DAPI staining, and they show you see it actively integrate during this S phase until before S phase, here, and it was very minimal. And then using this only they are able to track the Me1, Me2, Me3 localizations, which I show here. You can see the Me1, the distributions mostly within the nucleus obviously, but is small punctate, whereas Me2 you see the nuclear periphery. So, this also tells that each and every intermediate mark has its own functions, right? Otherwise, why it has to show the differential distribution? And coming to this Me3, you see predominant spotty localizations, real punctate. This is the condensed chromatin, repressed chromatin, or you can call heterochromatin. So, the because it's very intense, you can see with the DNA staining also. Now you can use it with the DAPI also, and the DAPI stains DNA in only DNA intensely, and then the intense regions within the nucleus also get intense blue color staining, which is matched with this color, with this K9 trimethylation also, which it tells that this has the function of gene repressions, or it distribute only to the repressive genes. So that's very clear, right? So the at least you should keep in mind Me2 mostly nuclear periphery. This is H3K9 Me2, and whereas Me3 the as a condensed chromatin. So this is basically heterochromatin. This directly tells us yes, this is gene repressive mark. Yes, so the chip sequencing, don't worry for these crowded peaks. Almost all these studies are confirmed across the various tissues and cell. Um, the mark is very poor. Sorry, the mark is mostly at the gene poor chromosomal regions. That means active genes wherever present, obviously this mark is absent, which makes sense because active genes need to be always have the activation mark. So almost all the heterochromatin are repressed genes having these typical uh, peaks, the chromatin enrichment peaks. I mean, this is a chip assay followed by sequencing, which makes sense because it is a connected to the gene repressive functions. And then they correlated with this mark and this mark. It almost similar. Okay, K9 Me3 mark and K4 H3K20 Me3 mark, and Histone 4 tail and 3 tail. In both the cases, the distribution more or less similar to the centromeric region. So the point here, the many reports have confirmed that both the repressive marks, many places in the genome coexist. That makes connection also, because both are repressive mark, but of course there is individual functional variations. Largely to repress the almost all the heterochromatin or pericentromeric heterochromatin, these marks works together. Okay, so I briefly introduced the K9 trimethylation marker, right? I showed the Me1, Me2, Me3 localization pattern. Now I'll explain the specific enzyme the way I explained to the H3K4. There are enzymes, specific enzymes, which methylates only at these positions. Because it's knowing a mark function is okay, you should also know about the epigenetic enzymes or epigenetic protein, because this is epigenetic machinery. That is why I try to introduce this enzyme name. We should aware about it. Okay, these are the enzymes known as a gene activation mark. How does it activates? By methylating so on some positions. These are the epigenetic enzymes or repressive function. How does it do? By methylating so on some positions. So the major enzymes in human is G9A and then SUV39H1. So these are the two proteins in human. It methylates at histone 3 lysine 9, and in a fungus it has been shown there is a protein which is kind of first characterized K9 methyltransferases from fungi, that is a DIM5 protein which methylates at histone 3 lysine 9. So here the comparison of these various substrate, the K4 substrate, 36 substrate, and histone 4, K20, K9, K27. These are major sites or methylation sites which I am going to explain, right? In all the sites when they compare it, the K9, K27, it is almost similar. Okay, but although it is similar motive, different enzymes. The SUV39H1 methylates only K9, not H3K27, although you have a same substrate, although you have same target substrate, same histone tail, but they're different. So that is how these epigenetic enzymes are getting regulated. Only selective enzyme, each and every positions, only specific set of enzymes are evolved to methylate and function as a repressive epigenetic enzymes. Right. So I'll just stop it now. We will continue the discussion about these enzymes. Two important enzymes which I'm going to explain along with the DIM. I'll introduce the DIM a little bit. Then I'm going to explain about these two proteins. There is a very unique behavior, a property between these two proteins. But again, only for a K9, H3K9 only. Okay. Then we'll move on to the another mark called 27. And also, I showed you the enrichment peak, chromatin enrichment peak for this Me2, Me3, which is largely present on the gene poor chromosomal regions. That means it is only repressed region, it highly enriched. And also I highlighted the K9 mark and then K20 and Histone 4 mark, trimethylation mark. Most of the cases coexist because both are repressive epigenetic methylation mark. Right. I will introduce the key epigenetic enzymes which introduce the methylation mark at these positions. The major enzyme is in human is ZUF39H1 and then G9A. This is in human and mouse, and the DIM is in a fungus Neurospora, because this was a kind of first enzyme. Its structure was available. The histone lysin methyltransferase structure was available, or the people made a structure for this protein, and that helped us to understand the mechanism of human methyltransferase as well. If you look at it, the substrate backbone for a K9, this one histone 3 tail, it is ARKS, and then histone 27 also. I mean histone 3 lysin 27 also ARKS. Of course, other target lysin, histone 4 lysin, this one flanks are different. 36 also flanks are different, and then of course the H1B 26, that is the conservation, but this is less important. But these two are repressive epigenetic positions. Histone 9, histone 3 lysin 9 and 27. Although it has a more or less similar flanks regions, but there is a enzyme preferences for this highly varies. For K9 positions, specific set of enzyme, in this case these two enzymes, and 27 I'll explain there is only one enzyme for this, which does not methylate K9 but only K27. Similar to the 27 and similar to the G9A and 39H1, it methylates only at K9 positions. Very rarely, under rare circumstances, it can methylate at 27 because of this substrate at in vitro condition. In vivo, there is no evidences. So that tells how this histone lysin methyltransferase are specific. Although it has a set domain, conserved catalytic pocket, but it has a diverse substrate specificity on histone itself. I mean, although lysin is a target amino acid, but doesn't methylate every lysins. Although there is a flanks also conserved. So that shows these enzymes are evolved with the specific functions, particularly these methyltransferase epigenetic enzymes. DIM5, I'll introduce. This is the identified from Neurospora crassa. It's a fungal eukaryote. When they were doing the mutational screening, and they found there is a strong correlation between this DIM5 and then DNA methyltransferases. So there is a two protein. DIM5 is a histone lysin methyltransferase in Neurospora, and DIM2 is the DNA methyltransferases. So you all know now DNA methyltransferase is a repressive epigenetic protein because it introduces the cytosine methylation and that function as a gene repressor mark. And then the DIM5, it is a histone lysin methyltransferases. So what they found in the very first in the study, when they knock out this protein histone lysin methyltransferase, they found the significant loss of DNA methylations. Surprising. When they knock out the histone lysin methyltransferases, they see loss of DNA methylations. Then they concluded, okay, so these two are different enzymes, but it works on a single loci or single positions. That means the identified DIM5 also may function as a repressive enzymes. Later only they characterize, okay, at which position it can methylate, and then they identified this can methylate at histone 3 lies in 9th positions. Now you can connect. This is also repressive functions. DNA M6 5 methyl 5 methyl cytosine 5 methyl cytosine also repressive functions. So there is a strong coordination between these two proteins. So this is how they identified in early days. You see, this is one of the approach I explained during the DNA epigenetic methods, restriction enzyme based assays. Just focus on the first picture. This is a wild type. So D and S is DPN2 digested and S is SAW3A1 digested. So the S enzyme mediated digestion is, the 5-methyl cytosine blocks the activity of this enzyme, whereas DPN2 particularly it can digest the enzymes. That's what it sees here. The wild type genomic DNA isolated, and when they subjected to this particular DPN2 enzyme, you see degradations. There is no DNA. Whereas when they treated with the SAW3, and then it cannot digest because pre-methylation is present. Focus only on wild type. That's why you see the appearance of these bands, right? Now, when they delete DIM5, they see the pattern similar to the DIM2. DIM2 is a DNA methyltransferase. Obviously, when you delete DNA methyltransferase, no DNA methylation. That is why you can see degradation here. But same is happening in the DIM5 as well. So that's why they concluded these two are function as a repressive enzyme. Later, the biochemical assay confirms this is histone lysine methyltransferase that lies in 9th positions on histone 3, and whereas this is a DNA methyltransferase. Right, this is what I said, the DIM5, the biochemical characterization, how they identified the histone lysine methylation activity. Here, they use the nucleosomes as a substrate, all 4 proteins, histone 3, 2A, 2B and histone 4. And then they conducted assay using the recombinant DIM5. How do they do it up? They use this nucleosomes plus DIM5, this is a substrate, an enzyme, and plus cofactor. Cofactor is SAM, and this is tritiated. Okay, isotope labeled. And then after methylations, and then they separate the protein, and then they do the autoradiography. This is autoradiography actually. And then when there is, wherever there is a methylation happen, you get this tritium signal, or this is the radioisotope signal. And then that shows the protein methylates only on a histone 3, not on H2, H2, H4. That is how they identified actually. So in the first case, they predicted it can methylate histone methyltransferases, but in the second experiment, they could narrow down, yes, it is specific to the histone 3. Then at which positions? The same assay one can think about it, or they have done it up, where and these the ARKS, this is the target 9th positions, can be mutated to ARAS. When you mutate to alanine, no target alanine, and then no methylation reactions. So this is how you will end up to identify the substrate for histone epigenetic enzymes. So this is how all they identified. So these are the different epigenetic methods they used up. Now we know, yes, this can methylate this position and this position, etc. and so on. But how do they identify it using these approaches? Even today, if you want to know or if you want to identify the PTT enzyme at which of the lysines it can methylate, this is the approach one has to think about it. Right. So I introduced the DIM. The reason for this, because the first histone lysine methyltransferase structure was available for the DIM5 protein, not even for a human or mouse or Drosophila proteins. And because this allowed us to study the mechanism of epigenetic enzyme to histone 3 lysine 9, very important repressive mark. Right. So during the structure identification, they identified very unique property for the protein. So unlike regular or canonical histone lysin methyltransferases, the DIM5 or which are other enzyme which I am going to explain, either SUV39H1 or G9A, these two are human proteins, keep in mind. These are a human protein which methylates at histone 3 lysin 9th. It has high activity at alkaline pH. Very surprising. And that's they identified from the structural studies, and then they found it is highly active when it is pH is around you see 8 plus. So when you want to conduct the assay for this particular enzymes, or when any methyltransferase enzyme which you hypothesis which can methylates at lysine 9th positions, better you should do assay at this above 8 plus. You will see hyper high activity. If you conduct a normal physiological condition, you do see activity, but you will have the very high activity at this condition. This is a kind of conserved mechanism across lysine 9 methyltransferase. Right. So again, see another property they identified. So after that only, they identified dimethyl specific enzyme, trimethyl specific enzyme and all. So the reason, okay, so I'll try to explain this, you'll understand. Here they conducted on assay the wild type enzyme using the histone 3 peptide, histone 3 lysine 9 flanks peptides, and then they mutated these F281 to tyrosine, aromatic amino acid only. So by changing this phenylalanine, this bulky aromatic amino acid to tyrosine, so basically they are changing the pocket, the catalytic pocket size. That is the only thing. What they identified, because of the changing this pocket, you could see in the wild type in a 5 minutes to within 30 minutes it can convert. This is the LCMS analysis. You have unmethylated peptide fragment, monomethylated one, dimethylated and trimethylated in 5 minutes. In 30 minutes, all are converted to the trimethylated. So this is the property of the lysine methyltransferase, particularly the DIM5 or lysine 9 methyltransferase. Whereas the mutant one, it is sluggish. Even 3 years, you see, the trimethylation level is very minimal. So what they're trying to infer is the methyl pocket is very, very important for a successful activity. When you change a methyl pocket little bit, although you have the same aromatic amino acids, but the conversion rate is decreased. And that was a key mechanism they identified, which helped for a scientist to identify histone 3 lysine 9 Me2 specific activity, and then histone 3 lysine 9 Me3 specific activity. That there are two different enzymes was identified later. But this was the initial assays was kind of given a kind of some kind of suggestions. Yes, the enzyme can behave when you alter the catalytic pocket a little bit. You see that this is a very clear evidence. This is a very strong data. And then same they tried with another enzyme SETS1, which actually methylates with the H3K9, and then they see the similar phenomenon. Right. So DIM5, it is in a neurospora. DIM5 also, DIM5 it is a histone 3 lysine 9. But coming to the human protein, that is initially it was identified in Drosophila, and the same protein is present in human. Human also it is ZUV39H1, but the name is from these Drosophila. The SUV is basically suppressor of variegation 3 to 9. You should read 39, not 39. Okay, homolog 1. So basically, this during the genetic screening, mutational screening in Drosophila, they identified this protein basically that function as a suppressor protein. That is what they named it. And later only they identified this as the Z domain, and then which has the catalytic arcade towards the H3 proteins. So the important discovery or important epigenetic protein discovered in the Drosophila, and then later it identified in the human as well. SUV is a very important epigenetic repressive proteins in the human cells. SUV39H1 is a very key enzyme which methylates at histone 3 lysine 9. More importantly, it produces Me3 mark. I'll tell you why it is Me3 mark. Yes, subsequently, the biochemical studies using this protein identified, it methylates at histone 3 proteins. You see, these are key authors. David Alice is a part of this discovery, is the editor, the book which I referred for your epigenetics, right? So those are the pioneers who showed these epigenetic activity for these various methyltransferases. And they use the classical biochemistry approach as I explained for this DIM. They used histone 3 substrate plus ZUF39H1, then cofactor SAM, and then they conducted assays. You see, it methylates here, the recombinant protein. So the very specific activity towards the H3 proteins. So they concluded, yes, it is also like DIM, this is also specific towards the H3. But later at K9 or not, they made a mutation on H3. This is wild type lysine, and then the mutant is, instead of lysine to alanine, you change the target lysine to alanine, and then you will see loss of methylation. Then that is your result. You see, when I change the target lysine, I see there is no activity. That means the enzyme is evolved to methylate only that particular patient. This is how you end up to identify is specific. These are the enzymes specific to these target lysines. Yes, these are all again biochemistry assays using the same strategy what I explained here. You can see very highly active only on H3, and then you can see here as well. When they give H3 alone is active, and then again all whole nucleosome has a structures, it will methylates only on histone 3 positions. Then during these biochemistry assays only they identified, I explained during one of the earlier class as well, the Histone phosphorylations, it function as an antagonistic signal for this repressive mark. If you recollect the nucleosome structure mechanisms when I explained, and this is lysine 9, and then this is A or KS sequence, right? So 10th positions, this is 10th and 9th positions. And when this serine 10 is phosphorylated, and then this does not allow to methylate at this position by this enzyme. And this is the biochemistry assay. How do they do it up? How do they identify mechanistically? This is the simple approach. They use these peptide carrying these phosphorylations. You see here, when the Histone phosphorylated peptides, zero activity, right? And here you see full activity on the without phosphorylations. That means when the Histone position is phosphorylated, that does not allow the enzyme ZUF9 to bind on the substrate itself or nucleosome itself. Very clear. And then they tried for the phosphorylation activity. Same is true. The Histone phosphorylation is abolished when it is a methylated. It is both are antagonistic functions. When methylation is present, Histone phosphorylation not allowed, and Histone phosphorylation is present, and then again methylation is not allowed. So this never coexists. That's the point. Right. So where is it localized this mark? I said this this protein, the protein methylates at histone 3 lysine 9, lysine 9, and then the localization study shows that you see these are all spotty localizations. Just typical to these condensed, this is the DNA staining, DAPI staining, condensed structure. So which clearly shows its protein function as a heterochromatic proteins or heterochromatic localizing proteins. Likely is going to introduce a repressive mark, right? Right. So how exactly with the, after localization they come with the model why it is present in the heterochromatic, because by the time they identified, we will see about this story later when I talk about epigenetic reader topic. The HP1 is a protein which bind to these methylated positions or the histone 3 lysine 9, and then these two are always coexist, and that is the reason it will form a spotty. So basically this is a heterochromatic protein and this is also heterochromatic protein because it methylates heterochromatic mark histone 3 lysine 9, and then when these two are form a complex, it helps to spread. That is a coordinations, positive feedback loop. which helps to spread the repressive mark nearly 100 kb in the heterochromatic regions. Imagine in the post-replicate regions, when post-replication, it has to spread the mark. How does it spread? Through these coordinations. Right. So, it's very clear ZUB39 is the repressive enzymes because it methylates at histone 3, lysine 9. Very clear. It is lysine 9 is a repressive mark. That means the nucleosome carrying this particular position mark is always a repressive. That gene is repressive and is inactive. So that means it is possible it can interact. People identified it will interact with the DNMTs. DNMTs is again repressive enzymes. HP1 also repressive proteins. MBD repressive. It all like activator complexes. The repressive complex also it forms as a full repressive complex you see here. All are forming a complexes, and then obviously it will interact with the H3K as well because this is also repressive. It removes the deacetylase. And along with the repressive functions, it highly essential to repress or maintain the pericentromeric heterochromatic regions. But basically the chromatin stability, chromosome stability is highly achieved by these enzymes by introducing the trimethylation marker, the histone 3 proteins. And coming to the biology, it is highly essential by default, you can imagine, because the knockout or deficient, it is severely impaired the viability of animals, particularly mouse. Studies shows that it is highly essential because the knockout animals shows the high chromosomal instabilities. So, which clearly stabilizes this is essential to maintain the chromosome shape as well by successful introduction of the trimethylation mark at the chromosomes. And because of the loss of the protein, it just increases with the turmerics possible because of the chromosomal instabilities. And then it is also linked with the hypogonadisms as well as the spermatogonic failure due to this SUV39H mutant or the knockouts. So it is crucial for these methylation at these pericentromeric regions, and which is essential to protect the genome. So altogether is very essential enzyme along with the gene repressive functions. It is essential to shape the chromosome structures. Right. The second enzyme, the first enzyme what I highlighted is SUV and second is gene. So these are two enzymes I am going to highlight in connection with the histone 3 lysine 9. But the key difference you have to keep in mind, the SUV 39H1, right, and then G9A. So this is the enzyme introduces at trimethylation, histone 3 lysine 9 Me3. That means the same enzyme can introduce mono, di, tri. Okay. Whereas this enzyme introduces methylations only di, histone 3 lysine 9 Me2. Very important. So the GNA is a dimethyltransferase. No, it never introduces the trimethylation in the genome. It always, the in vitro studies, when you incubate longer, it's possible it may produce a tri. Otherwise, this enzyme is only dimethylation at least on the same positions. It is possible that the G9A can introduce Me1 followed by Me2 on the nucleosome of the histone 3 lysine 9. Then this can be converted to Me3 by SUV. So the SUV39 is the trimethyl. That is why it is highly present on the repressed regions, right? Heterochromatic regions, because it introduces the trimethylation mark. So, but that means dimethylation is not repressive mark? No, this also repressive, but not heterochromatic mark. Okay, and the SUV is heterochromatic enzyme and GNA euchromatic enzyme. but it's repressive. In fact, few literature started using the term GNA is euchromatic histone lysine methyltransferase. Histone lysine methyltransferases. Euchromatic histone lysine methyltransferase. So shows that it is repressive for the euchromatic genes. So there are genes which need to be repressed in the euchromatinized regions. That places the GNA takes care of it, which introduces maximum dimethylation. So dimethylation also, that means repressive function. And the heterochromatinizations or heterochromatin mark is this ZUF93H1 mediated trimethylation mark. So these are the key differences you should keep in mind. Okay, so that's why I want to restrict only these. There are few other enzymes which can methylate, but these are the predominant epigenetic enzyme in connection with the histone 3 lysine 9. Okay, one is dimethyltransferase, other is trimethyltransferase. Both carry the set domains, both carry the same catalytic motif. This is all fine, almost similar, but the substrate preferences varies. That is why the mutation explain why these enzymes carry the differential substrate productions, right? But this protein has some additional domains, GNA, that is actually, it's called anchoring repeat regions. It is similar to the WDR. I explained for these histone 3 lysine 4 methyltransferases, right? Set1 or MLL complex ASH1. For this complex, this helps to convert di to tri, and here that sort of functions, but here it is also function as a protein docking site to recruit a many repressive proteins in the euchromatin regions. That is the function of this repeat region for the GNA proteins. So this is how these anchoring repeats looks like. You see the coil to coil structures, helix loop helix structures in the anchoring repeat, and it is an important site to recruit proteins for a repressions. Right. Okay. So now it's clear. You know about now ZUF39H1 and then GNA. To know a little bit more why it is the identified can introduce dimethylations. You see, this is the one of they've conducted on the process methylation assays for using this protein, and note down here they use histone 1 to 24 peptide as a substrate, and then here they use a DIM, and then this is the trimethylation mark. Over the time, the DIM, this is unmethylated. So unmethylated peptide levels comes down, and then trimethylated Me3 marks goes up when they use a DIM. When they use a G9A, and this was true with when they use a ZUF also. Okay, but in here they showed only with the DIM. The same is true with the SUV39H1. When they conducted assay, same assay with the G9A, you see the unmethylated peptide level comes down, but the elevation of only dimethylated peptides and Me3 slowly comes on. When they incubate really longer time, this may comes, but the major population is, so that means enzyme pocket is very important. I mean it's evolved with the small pocket, because the same enzyme, if you introduce a 3-methyl group, means the pocket should be in highly dynamic, right? Because monomethylation, then it should have a space to accommodate dimethylation, and then dimethylation becomes substrate to accommodate to convert the trimethylation. So that pocket plasticity is very important. Here that pocket is evolved only, it can accommodate 2 methyl groups. That was the mechanisms explained using the structural studies. Yes, this enzyme can introduce maximum only dimethylation. That was true. Later they identified, this was a mechanical information, and cellular level it is confirmed later. Yes, this is distributed only in euchromatin not in heterochromatin. That makes sense. The mark also present only in euchromatin. So whatever the mechanisms they identified in vitro, strongly matched with the in a cell as well. Right. So not only that by assay, but in cellular level also, when they knock out this particular enzyme, they see reduction on the dimethylation here. You see dimethylation reduction here. This is knockout cells. When they re-express gene NA, they see gain of methylations. And but whereas other marks are not affected, either phosphorylation, Histone phosphorylations, or K9 acetylations, or K4 dimethylation, Histone 3 K4. So all these mark not affected, whereas only these dimethylation or Histone 3 affected. This is using these antibody for Histone 3 lysine 9 Me2 antibody. Yes, so I said it's not only for in vitro assays, in the cellular level as well, this was true, because you see very clearly HPV1 I already explained briefly, and we will see in detail in the reading classes and epigenetic reader classes. It is a part of a complex with the SUV39H1, right? So that means it is all heterochromatic spotty localization. You see there is a spot localizations, whereas gene is not. That is why it is named as a euchromatic methyltransferases. You see, this is not spotted; it's distributed throughout nucleus. It's very clear. And then, whereas this is distributed only spot. By looking, this will be confusing for you, but just focus only on a SUV3 and H1. SUV3 and H1, this one, the HV1, you see the yellow spotty co-localization. Whereas G9A and then SUV does not localize. So the red is SUV spotty, and then the green is your G9A, which is kind of diffuse, although you have some spotty regions here and there, but does not co-locate. No yellow color here. That is a point. So these proteins within the cell not coexisted. That means it has a independent functions. So it connects very well. SUV39 is a trimethyltransferase for heterochromatic regions, and G9A is a dimethyltransferase for euchromatic. For euchromatic gene silencing is G9A, heterochromatic gene silencing is SUV39H1. Yes, these are the confirmatory assays when they knock out these embryonic stem cell for this G9A, and they could see these these loss of protein. You can see here these diffused localizations, and here loss of protein in this diffuse localization pattern here. Again, additional confirmations for GNA, ZUF39H1 in the localization pattern, and then you see these monomethylations and dimethylations patterns. It's more like diffuse here, here, and then trimethylation is spotty, which clearly shows that this is heterochromatic and this is euchromatic, and that's matching with the GNA localization as well. And then additional assays have confirmed that the loss of GNA in the cell, or a knockout of GNA in both the copies. That this means you have this plus minus. I mean, one allele is lost, one allele is retained. Here both allele, that is the meaning here. And you see only this dimethylation is lost, trimethylation is still present. Right, this was GNA is functional to introduce a trimethylation on a specific heterochromatic regions. It's very, all these assays are clearly established, because now we understand, now we know very well. Now from this we are keep asking question. It's function at various loci and all. But before that, how this has been localized, where it is present, who does, and all the informations by the basic science researchers identified. So by knowing this, you can build more questions and can gain a more knowledge from now onwards. So that's the whole idea I want to introduce. Okay, GNA function as a dimethyltransferase, SUV39 function as a trimethyltransferases. Right. Next, I will introduce very briefly to another protein. So, only two protein you keep in mind in connection with the histone 3 lysine 9. Okay. See, the one more protein which I'll introduce, it's called GLP. It's GNA like protein. It is another protein, G9A. It is a dimethyltransferase, and GLP also dimethyltransferase. It is later identified. It almost similar like TNMT3A 3B. Okay, in major cases works together. In fact, the studies shows that this protein may form a complex and then it function to introduce the dimethylation in the euchromatic genes. So, dimethylation itself sufficient to repress the genes in the euchromatic regions. Right. So, it is highly related to the GNA because the set domains and then many of the structures almost similar, not a very big differences between the GNA and GLP. And the nuclear distribution also, these proteins highly similar, and then the biochemical studies also confirms, and both are methylating at the histone 3 only. So, all the data confirms similar, and then more importantly, they found this protein coexist in most of these cells. But also the report have shown that need not to be every cell will express both the protein. The cell expressing both the protein will coexist. There are in some cell types only G9A. There are in some cell types only GLP. Okay, but both their function to introducing the dimethylation at the histone 3 lysine um, uh, 9. Okay, so now so far you understood the information about the GNA. It is a major euchromatic methyltransferases, right? At very rarely, again biochemistry studies, in vitro studies, it can methylate at 29, but at cellular level it's not very clear. That means it is a strict enzyme towards this K9, histone 3 lysine 9 methyltransferase, and it's essential for the gene silencing at euchromatic regions. So, which again, if you recollect the localization pattern what I showed, dimethyl and GNA, you all able to connect it. Still repressive functions, but euchromatic. Whereas these SUV39, it is strong spotty localization, that is the heterochromatic regions, which shows that trimethylation is essential to form a condensed chromatin structures.